Alright guys, we are transitioning onto something called protein synthesis. So as you recall from our cell energies unit, synthesis means to create or to make. And so back in that unit we were learning about photosynthesis. And so that's the process of using light in plants to create a usable form of energy. So now we're talking about protein synthesis, so it's the creation of proteins within our cells. And so ultimately, DNA is going to be our starting point, and protein is going to be our final goal. So another way to say this, and what we're going to learn about is called the central dogma of genetics. So my guess is a lot of you are excited because it sounds like this is going to be all about dogs. Well, unfortunately, this has nothing to do with dogs, but it's going to be important to remember the phrase central dogma because it's going to come up a lot in the near future. So, as you recall, DNA is the blueprint for our genetic information, our cells, our entire body plan. And so all of our information is held within these strands of DNA. And the DNA is in the nucleus. Unfortunately, the DNA never can leave the nucleus. If DNA is leaving the nucleus, something catastrophic is probably going on and the cell is probably dying or dead. So you can look at this kind of like a library. There's a ton of information in the nucleus, just like a ton of books. But in this library, you can't check out the books. The information cannot leave the nucleus. So we need to come up with some way to get this information out because proteins are made in ribosomes, and ribosomes are in the cytoplasm, which is outside of the nucleus. So this is where protein synthesis comes in. So there's two primary steps to this. Step number one is the transition from DNA to RNA. This process is called transcription. The process between RNA to protein is called translation. So there are many types of RNA out there, but we're going to focus on three primary types. So the first one is called mRNA or messenger RNA. So that letter in front of the RNA is always going to be your indicator for which type of RNA we're talking about. So mRNA is kind of like our UPS or our FedEx driver of this process. It copies the information from the DNA, directly from the DNA, and then carries that copy out of the nucleus into the cytoplasm towards the ribosome. Next, we have tRNA or transfer RNA. To the right, this is kind of our blue structure that's right in the middle. And so this carries our amino acids to the ribosome and the mRNA. So if you notice on the very top, that black circle, that's going to be our amino acid that the tRNA is carrying. And our last one is going to be rRNA or ribosomal RNA. This RNA is ultimately the building blocks for ribosomes, so it creates the subunits that come together to form a ribosome. So again, the whole purpose of transcription is to carry this information from the DNA out of the nucleus towards the ribosomes. So again, the majority of this takes place inside of the nucleus. Obviously, the end is taking place in the cytoplasm, but the majority of transcription takes place in the nucleus. We're starting with our DNA, and our end goal is the creation of mRNA. So here are the steps, I guess the basic outline of transcription. So similar to DNA replication, the gene or the DNA needs to get unzipped. So if you look below, the purple strands are going to be our DNA, and we can tell because thymines are still present in orange. And the mRNA is going to be the red strand below. And again, we can tell it's RNA because the thymines are replaced with our purple uracils. So the mRNA ultimately forms a complementary strand for the section of DNA we're trying to copy. So all of our base pairing rules that we learned in DNA still apply. So after the complete mRNA strand is created, it detaches from the DNA and is released towards the outer portion of the nucleus to enter the cytoplasm. Then the DNA zips back up and continues on its regular functions, and the mRNA leaves the nucleus. So this is another way to look at it. So on the left, we have our DNA completely zipped up. Then what happens? The DNA unzips, and the complementary mRNA strand attaches to the DNA. Once the mRNA strand is complete, it detaches, leaves the nucleus, and the DNA zips back up. Alright, let's see if we can try to form the correct mRNA sequence for this DNA sequence. So go ahead, try and match up the correct letters, remembering that it is mRNA. So take a second, try and do it. Alright, hopefully our mRNA sequence looks something like this, remembering that our T's are still going to match up with our A's, but our A's are going to match up with uracil. Right, so that's our big difference that we're looking for.